Hi everyone, welcome to lesson 2.6, enzyme specificity. Make sure to fill in your notes and keep up with your vocab as we go through. So here are a couple of vocab terms from last class when we introduced enzymes. So recall, okay, we have some chemical reactions essential to life happen so slowly or require such high activation energies that they could never take place on their own. So the catalyst, and then we had enzymes, and enzymes are proteins that act as biological catalysts. The role of enzymes was to speed up the chemical reactions that take place in cells. And then we went through this energy diagram last time, which if you remember, we had the blue line was the reaction without an enzyme, the red line was the reaction with an enzyme, and we saw that the reaction with an enzyme had lower activation energy. And because of that, that reaction proceeds at a much faster rate. So last time we looked at two examples of enzymes, catalase and carbonic anhydrase. And then here are some more examples of enzymes, particularly those that are involved in digestion. So please note you don't have to memorize this. This is just looking at a couple examples. So the first thing to notice is the names of the enzymes. So notice how majority of them end in this ACE, A-S-E. There are some enzymes which have been around longer and they have older names that don't end in ACE. So a couple examples, pepsin, trypsin, both of which are on here, chymotrypsin, all those are enzymes also in the digestive tract, but pretty much all other enzymes end in ACE, A-S-E. So if you see something like polymerase, lipase, etc., you know it's an enzyme just by its name. Now the second thing to notice is there are many different digestive enzymes, okay, all listed here. And look at the role in digestion. Notice how each has a specific job that is slightly different than the others. So that brings us to kind of what we're talking about today for enzyme specificity. So enzymes provide a site where the reactants or substrates can be brought together to react, and this site reduces the energy needed for the reaction. The substrate is the reactant of an enzyme-catalyzed reaction, and enzymes act only on specific substrates. So for example, amylase, an enzyme in your saliva, assists in the breakdown of starches in your food. Okay, and then just like on that last slide, enzymes end with ASE, so we can figure out something is an enzyme even if we've never heard it before, like amylase. So an enzyme's activity is determined by the shape of the enzyme, as seen in the image below. Okay, so here is a rough picture of an enzyme. Okay, and then here, this little red piece is the substrate. Okay, and then here in this next photo, we see the substrate fits very well with this enzyme. Okay, now what is this place called? That is what we call the active site. So typically an enzyme is a large protein with one or more deep folds on the surface. The substrates bind to a site on the enzyme called the active site. So in this case, this kind of little divot here, that is the active site, and that is the part of the enzyme where the substrate binds. The active site and the substrates have complementary shapes. So notice the substrate, this red piece here, the thing that's going to react with the enzyme looks like it will fit well in this active site. Again, because they have complementary shapes and that fit is very precise. So let's go back and look at carbonic anhydrase. So we talked about this in the last class, and all of this information we went over before, but now we're going to kind of look at this reaction a little bit closer. So here is a diagram that we're going to go through. All right, so first off, these substrates are water and carbon dioxide. Okay, and again, these substrates are basically like the reactant or that thing that the enzyme's going to break down or bring together or act on. The enzyme itself here, okay, was carbonic anhydrase, and again, notice that ASE. The enzyme substrate complex is basically whenever these two things join. So you have your enzyme, your active site, which fits the substrates nicely, and that forms this enzyme substrate complex. And that is a temporary molecule formed when the substrate binds to the active site of the enzyme. So as it binds, okay, the enzyme does its job, whether that's breaking something down, building it up, whatever its specific job is. So as it does that, these substrates are converted into the desired products. The products are released, which in this case was carbonic acid, okay, and then the enzyme can go and do its job again, right? It can go and break down more products or join more reactants together, depending on its job. So here's a check for understanding looking at a similar diagram on that last page. So go ahead, pause here, read through this diagram, and try it on your own. Okay, so the reactant or substrate, in this case it was labeled, okay, so it kind of helped you, but it is sucrose, okay? What are the products? So we're going to kind of fast forward through this diagram, and we notice that the products, step 3, glucose and fructose. So sucrose in this case was broken down into glucose and fructose. Now, what type of reaction is occurring? So this one kind of goes back to lesson 2.4, and that is hydrolysis. Okay, so this one might be kind of confusing, but here's an easy way to remember that. Remember sucrose, okay, that those two sugars join together, we're going into the reaction, and then you notice water is added here, and then something's broken apart, right? So remember, water added, breaking apart, that is hydrolysis. What type of molecule is sucrose? So sucrose, if you recall, was a disaccharide, okay, or a carbohydrate. And what types of molecules are glucose and fructose? Again, those were both carbohydrates, but specifically they were monosaccharides, okay? So again, looking at this diagram, just kind of make sure you understand what's going on. The substrate binds, it forms that enzyme substrate complex. That enzyme does its job, which in this specific case was to break down the substrate into the products glucose and fructose. Those are released, and then it says the active site is available for another molecule of substrate. So then the enzyme can go and do its job again. So how do substrates bind to enzymes? And we have a couple models that answer that question. So the first one is called the lock and key model, and that's a model of enzyme-substrate interaction that states enzymes and substrates have corresponding shapes, kind of like a lock and key, kind of like visualized in this picture here. So this is now considered an older model of how enzymes work, and we now know that the matching between an enzyme's active site and the substrate isn't exactly like two puzzle pieces fitting together. So that brings us to the induced fit model. And that is a model for enzyme-substrate interaction where the proper substrate is capable of inducing proper alignment of the active site, which will enable the enzyme to perform its catalytic function. So it just suggests that the active site continues to change slightly until the substrate is completely bound to it, at which point the final shape and charge is determined. So let's look at this induced fit in a little bit more detail, okay? So here's a good diagram. We have the substrate entering the active site of the enzyme, okay? So substrate, active site, this is the enzyme itself, okay? It binds, and then we get that enzyme substrate complex, or those just two things bound together. And then it says the enzyme changes shape slightly as the substrate binds. So right here kind of wraps around it just a little bit better. The enzyme product complex, okay, meaning like it is now completely bound to the enzymes and doing its job of breaking those apart. And the products leave the active site of the enzyme, and then that enzyme is freed up to go back and break down more substrates. So now we get to that question, how exactly do enzymes lower the activation energy? Well, here are a lot more details on how exactly the enzymes work. You don't need to worry about memorizing this information because, as you can see, there are many different ways they can speed up reactions. The main point is that enzymes provide a site where the reactants or substrates can be brought together to react, and that site is what reduces the energy needed for the reaction. Okay, so again, here, some more details on exactly how they can do that, very different ways. But just remember, enzymes provide a site where the reactants or substrates can be brought together, and that site reduces the energy needed for the reaction to start. So here's our first check for understanding. Go ahead, pause here, and try to answer these on your own. Okay, so let's check ourselves. So the lock and key model suggested that the fit between the enzyme and substrate is exactly precise, whereas the induced fit model suggests that an enzyme changes shape slightly when it bound to the substrate, and that resulted in an exact fit. So the induced fit model is the more widely accepted one today. The enzyme substrate complex was a temporary molecule formed when the substrate binds to the active site of the enzyme. Do all enzymes act on all substrates? That's that key word, all. No. Remember, enzymes act only on specific substrates. Now, whether that is only one substrate versus like 2, 3, 4, 5, that just depends on the enzyme, okay? But at the end of the day, enzymes act only on specific substrates. What is the energy needed to get a reaction started? And that was called activation energy. So now we're going to look at enzyme regulation. So enzymes can be regulated by other molecules that either increase or reduce their activity. Molecules that increase activity are called activators, and the ones that decrease activity are called inhibitors. So enzyme function can be inhibited, which means restrained or regulated, in two main ways. So here we have some vocab terms. Competitive inhibition, and that is when molecules sit in the active site of the enzyme and prevent the entry of the substrate. This is called competitive inhibition because the inhibitor competes with the substrate for the enzyme. Non-competitive inhibition, molecules do not sit in the active site, but they will attach to an allosteric site and change the shape of the enzyme. So that ultimately will change the shape of the active site, and the enzyme is generally less compatible with its substrate because of that. The allosteric site is a place on an enzyme where a molecule that is not a substrate may bind. That results in a change to the shape of the active site and the enzyme. So let's look at inhibition in a little bit more detail. Okay, so here we have two diagrams that kind of show the same thing in just two different ways. Okay, so down here we have non-competitive inhibition. Okay, and up top we have competitive here, and this one shows a normal reaction. So just kind of pause here and examine these diagrams on your own. So the main idea is that competitive inhibition completely blocks the active site, okay, as shown here, all right, and here, right? Competitive inhibition completely blocks the active site, okay, while non-competitive inhibition reduces enzyme activity due to the substrate bonding to an allosteric site, okay? So if you see non-competitive inhibition, you need to think of the term allosteric. Okay, so here is one example of how non-competitive inhibition can work, and here is another one. Okay, so the inhibition said to be non-competitive if the inhibitor and substrate could both be bound at the same time. So the active site itself was not blocked. The inhibition could result in the substrate completely not being able to bind, for example down here, or up here, the substrate can still bind, but the enzyme reaction could be blocked, okay? But non-competitive inhibition, both ways involve the binding on an allosteric site, which is going to change the shape of the enzyme. So now we're going to look at more regulation of enzyme activity. So remember, enzymes play essential roles in controlling chemical pathways, making materials that cells need, releasing energy, and transferring information. Because they are catalysts for reactions, enzymes can be affected in a variety of ways. Okay, so temperature, pH, and regulatory molecules can affect the activity of enzymes. So we already looked at competitive and non-competitive inhibition, so we're going to look at temperature, pH, and the amount of substrate and enzyme present. So here we have temperature. Enzymes operate most efficiently within a certain range of temperatures, and the majority of enzymes in humans generally work best at temperatures close to natural human temperatures, right? So temperatures outside this range break some bonds in the enzyme, which changes the shape, and then that's going to affect the functionality of the enzyme. So looking at this graph here, okay, we have rate of enzyme activity here, and then we have temperature on this axis. So lower temperatures cause the enzyme to be inactive, okay? And then here we reach what we call the optimum temperature, okay? And that shows us our maximum rate of enzyme activity. Once we get past that optimum range, if we get into temperatures that are too high, you notice this steep drop in enzyme activity all the way back down to 0. So when temperatures get too high, the enzyme becomes what we call denatured, and that will be a useful word for you to know and use. And denatured just means it's losing its shape and therefore functionality. So our next one, we're going to look at the pH range. So the pH scale down here indicates the concentration of hydrogen ions in a solution, and this concentration is how we determine if something is acidic or basic. The lower the pH, the greater the acidity. So anything basically pH less than 7 would be considered acidic. The higher the pH, the more basic the solution. So anything pH 7 and above is basic. Okay, now neutral pH right here is 7, okay, neutral pH, and pure water is an example of pH of 7. Not the water that you typically get in water fountains, but pure water. So for example, the stomach enzyme pepsin, which begins protein digestion, works best under acidic conditions, because if we go down here to where we find gastric juices have a pH of about 2. Okay, those enzymes are adapted to function best in this acidic environment. So imagine if that pH, for example, in your stomach went all the way up to like pH of 11. First off, that'd be very bad and you probably would not survive. But second, that enzyme would no longer function, okay, because it functions best within this low pH range. And then here is some extra information on acids and bases and buffers. You don't need to know this at this time, okay, but here are the definitions. Buffers are something that can help the body prevent sudden changes from taking place, and they play an important role in maintaining homeostasis. So our last two are the amounts of substrates and enzymes present, and those can also influence the rate of enzymes reaction or rate of reaction. So the enzyme concentration tells us increasing enzyme concentration will speed up the reaction as long as there's substrate available to bind. Once all the substrate is bound, that reaction will no longer speed up because there's going to be nothing for additional enzymes to bind to. The substrate concentration works in a really similar way. If you increase the concentration, it's also going to increase the rate of the reaction to a certain point. Once all the enzymes have bound, any substrate increase will have no effect on the rate, as the available enzymes will be saturated and working at their maximum rate. So here again we have this graph, rate of reaction, substrate concentration. Okay, so here we have that enzyme working, increasing substrate concentration. Once we reach this X, this point of saturation, the increase in concentration does not affect the reaction rate. Okay, and again, that just happens as all the available enzymes will be saturated, and therefore they're already working at their maximum rate. So we reached our exit ticket. Go ahead, pause here, and try to answer these on your own. So here's our exit ticket. Okay, 3 factors that affect enzyme activity are temperature, pH, and the amount of enzyme or the amount of substrate. What occurs during non-competitive inhibition? Molecules do not sit in the active site, but rather they will attach to an allosteric site and they will change the shape of the enzyme. The enzyme is now less compatible with its substrate. Which pH is considered neutral? A pH of 7 is neutral. A substance with a pH of 3 would be considered an acid. And what happens to an enzyme's rate of reaction when you increase the substrate concentration? Or you could also do this with enzyme concentration as well. So increasing the substrate concentration also increases the rate of reaction to a certain point, but once it reaches that what we call point of saturation, the enzymes will not have any additional effect, right? Because any substrate increase will have no effect because all of them are already bound together. So that is the end of lesson 2.6. Thank you for watching. Make sure to go back and check your notes and vocab and reach out with any questions.